So welcome everybody at home or in their offices to this webinar on oxidative stress. I'd like to start by introducing my co-chair, Sandra Lindstedt. She's a thorax surgeon from Lund University, and she's also a board member of ACTORS. That's a regenerative medicine section of ESOT. I'm Martin Hooglaan. I'm a biologist from Erasmus Medical Center in the Netherlands, and I'm also part of the ACTORS board. So this webinar is actually leading up to the transplant learning journey, and in the transplant learning journey, there's a symposium from the Basic Science Committee together with the VCA and ACTORS committees. So this will all be online, and you're encouraged to register for it. So the dates are the 15th to 17th of November. And the Basic Science Symposium is on the 17th of November. Before I hand over to Sandra, I have one comment. So we're going to have two very interesting presentations. And during the presentations, you can post your questions. At the bottom of the screen, there's a Q&A button which you can press. And then at the end of the two presentations, Sandra and I will read out the questions and we will discuss them. So please, Sandra.
Thank you, Martin. So this session on oxidative stress and how it can be targeted. For this session, we have two excellent speakers today that will enlighten us on this matter on oxidative stress and how it can be targeted. And the first speaker is Professor Jan Lindeman, and he's from the Department of Vascular Surgery and Transplantation Center, Leiden University Hospital in the Netherlands. And Professor Lindeman's research focus is on dynamics on human vascular disease with a strong emphasis on ischemia reperfusion injury. And his group has its expertise in working on human tissue samples, small proof of concept studies with advanced molecular readouts, but also on phase 3 clinical trials. So very welcome, Professor Jan Lindeman.
Well, thank you so much, Sandra, and also Martin, for this opportunity. I'm particularly grateful for the fact that the organizing committee did not pull back when I said that my research on oxidative stress is actually going into the direction that oxidative stress might not be involved in human ischemia reperfusion injury. And having just said this, I'm hoping that my research data is convincing to you and that it will be the basis for interesting discussions in the Q&A sections later on. So, having said this, there is a massive amount of data available on ischemia reperfusion injury, and we are actually doing a perfect job when it comes to preclinical research. The major problem actually is in the translation. We have done research now for 30 or 40 years, and we haven't made any progress. We spent billions of dollars, came up with, well, hundreds, thousands of potential therapies, which all proved to be very effective in the models, but not in the humans. And that also accounts for kidney transplantation. We still have to come a long way to reach the clinic with our therapies. And it was quite a while ago, I think 2006, that my colleague Sandra Schaapheder and I came up with the idea of using arterial venous differences to understand ischemia reperfusion injury. I had a long-standing interest in oxygen radicals. I think this is my first paper from 1989. And back in 2006, we realized actually nothing had changed. So our thoughts were that if we started measuring arterial venous differences over the kidney in time, that this would provide us with an image of the processes involved in ischemia reperfusion injury. So rather naively, we thought in the first sampling there would be reactive oxygen species, and then later on in the progress that we would see secondary and tertiary effects. Well, this is how it looks in practice. So we cannulated the side branch of the renal vein in the graft and positioned a catheter in the artery. And then over time, in the first half hour of reperfusion, we sampled the venous blood, shown over here with the low oxygen saturation, and arterial blood shown here. And the difference between the two is the absorption of oxygen by the kidney graft. Well, this is a proof of principle. And how does this look during the actual transplantation? Well, one of the first studies done by Dorothea de Vries, one of the PhD students at that time, showed a remarkable release of IL-6 from the graft. And that's shown
Sorry,
here.
Jan, your slides are not in view. Can you?
Let me. And haven't they been in? Are they in view now?
Yes, they're coming now. Yeah, perfect.
So I go back one slide, showing the oxygen saturation. So showing the arterial blood with a 100% oxygen saturation and the venous blood. And the difference between the two is the oxygen absorbed by the kidney. So when we looked into more detail in the processes associated with ischemia reperfusion injury, one of the first findings we noticed was remarkable release of IL-6 from the graft. And that's shown in this graph with the arterial IL-6 concentrations at the bottom and the venous IL-6 levels on the top graph. And what's showing this is that immediately following reperfusion, there is a quite substantial release of IL-6 from the graft. The next question was obviously, is this IL-6 beneficial, or is this actually part of the recovery process? So what Dorothea and Sandro did, they performed a mouse ischemia reperfusion model with clamping of the kidney pedicle, followed by reperfusion. And much to our surprise, we noticed that when we neutralized IL-6, that there was actually a much stronger injury. So in the context of ischemia reperfusion injury, in the acute phase, IL-6 appears to be preventive and limiting the damage. So what about oxidative damage? This is what we're talking about tonight. Well, we studied that in two clinical contexts. We studied kidney transplant, but also we studied cardiac ischemia reperfusion during mitral valve surgery with cross-clamping of the aorta. And in the over the heart, we positioned the catheter in the coronary sinus. So we were able to collect arterial venous blood over the heart for 24 hours. Well, what were the results of these measurements? Well, we looked at several markers of ischemia reperfusion injury, like MDA, but also isoprostanes and nitrotyrosine. And what's showing here are the arterial venous differences for the isoprostanes. And actually, there is not much happening at all. We actually don't see any signs of oxidative damage, neither for the kidney nor for the heart during the first half hour of ischemia reperfusion. And does it exclude oxidative stress and oxidative, well, it excludes oxidative damage, but does this exclude oxidative stress? Actually, no. Because if we have a competent antioxidant system, this can well quench all the radicals formed. So we looked into more detail, and we looked into the antioxidant system. And there are 3 systems actually in the humans. That's the antioxidant enzymes like superoxide dismutase, iron chelation, but the other system are the sacrificial antioxidants. They quench a free radical and are transformed in a stable molecule. And one of those antioxidants is uric acid. And when uric acid reacts with a free radical like superoxide, allantoin is formed. And this is actually a terminal end product in humans. So it's stable. So we also looked into allantoin, and we didn't see any allantoin formation at all, neither in living donors, in deceased donors without DGF, but also not in the donor grafts which later turned out to have DGF. So that's what we consider ischemia reperfusion injury. So summarizing this data, we don't have any evidence for oxidative stress in the context of ischemia reperfusion and ischemia reperfusion injury. So the question is, what's actually driving clinical ischemia reperfusion injury? And that's when we use delayed graft function as a readout for clinical ischemia reperfusion injury. And we used a quite robust definition of DGF with requirement of multiple dialysis and a duration of at least 7 days to exclude dialysis for hyperpotassiemia and other non-ischemia reperfusion related causes. So what we did, we performed messenger RNA profiling on biopsies taken just before reperfusion and 45 minutes after reperfusion. And what's shown here are 3 donor groups. So the blue, it's the living donors. In green, the diseased donors without DGF. And in red, the donors with DGF. And we had a rather paradoxical finding, which the first thing which is very notable is the activation of the redox response pathway, the NRF2 pathway, which is a very agile pathway. And much to our surprise, the highest response was in the living, and the lowest response was in the grafts with ischemia reperfusion injury. So the grafts which sustained the most injury and had the most stress. Another finding was when we looked at all the pathways popping up, most of the pathways had to do with metabolism. So keeping this in mind, we start wondering, isn't it about energy? Isn't the failing response in the grafts with DGF due to a lack of energy, simply because translation and transcription cost a lot of energy and ATP? So the question arose is whether ischemia reperfusion injury results from energy issues. So what we did, we looked into lactate as a first readout of metabolic competence. And what's immediately apparent is that if you reperfuse a graft, there is a washout of lactate in the first minutes following reperfusion. And in a living donor graft, within 3 minutes, there is metabolic competence. It takes a little longer in diseased donor grafts, but after 10, 15 minutes, there is metabolic competence. But this is not happening in grafts with ischemia reperfusion injury. So there is a failing oxidative phosphorylation, persistent glycolysis in grafts which later on turn out to present with DGF. So is there an energy problem? So we looked more into death, and we looked into ATP catabolism. And when ADP is not regenerated well from ADP, it's finally broken down to hypoxanthine. So we looked into hypoxanthine and again found a washout of hypoxanthine produced during the cold storage, and a rapid recovery of the ATP shown here by the disappearance of hypoxanthine release, but a persistent hypoxanthine release in grafts with DGF. And the reason for the decline over time is probably the exhaustion of the ATP pool. So this shows that DGF is associated with a profound metabolic collapse and the kidney not able to maintain its energy requirements. And that's further confirmed by the fact that we had a signal coming from plasmalogens, so the breakdown product of phospholipids. And the activation of phospholipids is linked to a profound ATP deficiency. So we have quite strong evidence that ischemia reperfusion injury relates to a metabolic deficit following reperfusion. And when we created the whole metabolome of ischemia reperfusion injury, we had a quite complex image on first glance, but actually it's not. What it's showing that there is ATP catabolism, so ATP is not generated well, and in response, the kidney is activating all catabolic pathways, so glycolysis, beta oxidation, glutamine oxidation, and autophagia. And this results in a redox stress because all the NAD is converted into NADH. So rather than clinical ischemia reperfusion injury being associated with oxidative stress, it appears to be associated with redox stress. So our conclusions are that in the clinical context, we do not have indications for oxidative stress as a driver of clinical ischemia reperfusion injury. On the contrary, it appears to be driven by an exhaustion of the high energy ATP pool with compensatory activation of catabolic pathways that may result in redox stress. These findings clearly contrast with that for the available animal models, such as the Ciucciani paper in Nature, who showed an inverse association between succinate levels and ischemia time, and a very transient activation of oxidative stress following by metabolic competence. But when we look into our clinical data, we found the reverse. So succinate level in kidney transplantation, succinate levels are inversely correlated with the ischemia times. So in contrast to the findings from Ciucciani. And the other aspect is that when you look into the kidney, the actual problem is on the level of the alpha-ketoglutarate, the hydrogenase complex, rather than on the level of succinates. So this is sharply contrasting with the data from the experimental models. So that brings me to the end of my presentation. Is oxidative stress a non-issue in clinical organ transplantation? No, it's definitely a problem, particularly now we are pumping organs. And one of the things we noticed during normal machine perfusion in studies done by Asel and Jason in our department was the massive accumulation of free heme released from the red blood cells used. And free heme is a very pro-oxidant molecule and highly toxic. And under normal conditions in our body, those transition metals are captured by haptoglobin, hemopexin, and transferrin, and they are currently not included in our reperfusion protocols. So I think this poses a major challenge for us for the future when we are going into prolonged pumping, particularly in the context of organ regeneration and organ testing. And with that, I would like to end my talk not without expressing my great gratitude to all who participate in this project, in this journey. And I hope to see a couple of questions in the Q&A session later on. Well, thank you so much. And I'm curious to hear Philip's talk.
So thank you very much for a very interesting talk, Professor Lindemann. And I really like the connection to the open heart surgery and mitral valve surgery with the ATP depletion. And I will definitely have some questions for you, but I think we take that afterwards. So thank you very much. Now we're going to the next speaker, and that is Professor Philip Dukowski. And Professor Philip Dukowski, he's a professor of surgery at the University of Zurich in Switzerland. And he's abdominal and transplant surgeon. And since 2009, he's been actually the head of the abdominal organ transplantation in Zurich. And in 2004, he joined the group of Pierre-Alain Colavin in Zurich, and he succeeded to translate basic research on machine liver perfusion into clinical application. And consequently, he did the first worldwide clinical application of hypothermic oxygenated perfusion in human DCD livers, and that was implemented in Zurich in 2012. So very welcome, Professor Philip Dukowski.
Yeah, thank you very much. I'm very happy to speak here. You hear me? Is that okay? You can hear me?
Yes, we can hear you.
Good.
We can hear you.
So I try to start my presentation. Do you see it? You see it?
Not yet, no.
Not yet. Okay, I have to check here. 1 moment. Yeah, where is that? Yeah, okay. Now it should come.
Perfect.
Yeah. So now you see it, right?
Yes.
Good.
Perfect.
So I'm very happy to say here something about current status of ex situ liver perfusion. I'm afraid I will say the complete opposite of the previous speaker, but very interesting, although, and I think it's a good point to discuss it. So I cannot not say everything about liver perfusion because I would speak for probably weeks. I can just put some small things and try to say something from the surgical side. And so I just start with these slides. You can share, of course, and you see now the two perfusion principles, which are now, I think, currently in the world competing, or at least done by different teams. So one side is on the left side, you see the liver with the organox system perfused with nice blood. The target is to have a most physiologic environment to let the liver sit like it is in the human body. So you connect, of course, the bile duct and you measure the bile. The liver is nicely producing bile. You can measure also lactate. You can measure all the things which we measure on the ICU, which is not much, by the way, but you can see something. And the opposite is on the right side. So that's liver which is perfused at 10 degrees Celsius. And it is not physiologic. It is swimming in water or in the fluid, and there's no blood. There's nothing which you can see. So that's an unphysiologic environment. And the question is, can we use both, or is something better, or what's about oxidative stress if you do like this? But this is only the procedure. We have also to decide which treatment would we choose as a surgeon, as a liver surgeon. This is a talk completely on livers, I have to say. So one choice is reduce the period of ischemia, which is, I guess, always there in the transplant business, more or less, in DCT more, of course. And the other side is prevent ischemia reperfusion injury, which I think clearly exists. And if you go for the left side, this is a treatment which is required at the donor site. So in principle, you have to introduce at the donor site the machines, or you decide to treat livers or also kidneys and hearts at the recipient center. That means you do the conventional harvest, the conventional procurement, ice box, you travel with the livers or with the organs on ice and treat at the center. So that is, of course, a different procedure, and your choice of the machine is depending on this. Because if you decide to reduce the period of ischemia, for example, by ESOT, that is ischemia-free organ transplant, that means connecting of the liver in a complicated machine directly in the donor and keep it on the machine until implantation, or you do NRP, normothermic regional perfusion, that's ECMO in donor, connect the donor to an oxygenated system, or you do the harvest and plug the liver immediately on a perfusion machine and travel with the machine, livers, kidneys on the machine, or you do the different thing, kind of avoid, repair strategy. You keep the cold storage and put livers after a previous period of ischemia on machines. That is an end ischemic approach, and I'm focusing here in this talk, because the talk is about ischemia reperfusion. I focus on the end ischemic principle in machine perfusion. So in principle, we talk then about addressing two types of injury. One is, which Jan always also said, it's immediate. It's always an immediate decrease or loss of energy products if we have no oxygen on board. That is depending on the ATP breakdown. And the other is the secondary oxidative stress during reperfusion, which is, if we listen to the previous speaker, of course questionable, but I think it happens if I look at our data. So two things: immediate decrease of energy products and secondary oxidative stress during reperfusion injury because the introduction of oxygen is necessary, but causes injury, which you will see in a second. And I think we should target, if we speak about perfusion things, we should target on both issues. And this is now the already mentioned Kuchani paper, and I think it is, at least in mice, that is studies on mice, which of course have to be repeated in human, but I show you some data in human. So lack of oxygen causes an accumulation of metabolites. I think that is clear. So it's not only succinate, which is here accumulating, it's also NADH or other metabolites from the citric acid cycle. So there's a lot of data available about that. Also hypoxanthine, of course, also lactate and xanthine, but there's no doubt that we have an accumulation of citric acid metabolites. And the surprising thing, at least for a surgeon, is that happens in many species, not only in human, that's in rats, in pigs, in other species, and it's also happening in many tissues. So we speak about succinate accumulation in lungs, in brain, in livers, in kidneys, and all of these metabolites, they change in the same way. That's the surprising thing for me. So you see here the red circled areas. This is succinate and also the purine metabolites. They go in the same direction in every species. And they are clearly depending, this accumulation is clearly depending on the time of warm ischemia. So if there's small amount of warm ischemia, there's of course less accumulation. If there's high amount of warm ischemia, like in DCD plus cold storage, there's a huge accumulation of succinate. At least we measure that always in human livers, by the way. So I think there's no doubt that we have this kind of metabolites, and the question is, why does it hurt? what's the matter about that? And we speak here immediately about something which I believe is central. If we speak about ischemia reperfusion injury, we speak about mitochondrial injury. Because, in fact, if you introduce oxygen to ischemic tissues, there's something happening which is called reverse electron transfer. So the electrons are not driving forward in the direction of the ATP synthase. They are driving actually backwards to complex 1, and they are causing here injury. And that is the cause, I guess, the primary cause of oxidative stress. And that is happening in the first minute. It is not happening after 10 minutes, not after 20 minutes. That is happening in seconds, and it's happening in mitochondria. And of course, that is very difficult to measure. So it may happen that you don't capture that if you don't look inside mitochondria. And just putting here some explanations how a surgeon can, I guess, how a surgeon can understand what's happening about in ischemia reperfusion injury. So the mitochondria is this wheel, and we have normally the physiological electron flow that is like, if I may say so, like water which is going down to the wheel and driving the wheel in the forward direction, producing energy. So if there's no oxygen, it's obviously a stop of the water, and we have no energy. But there's also something happening. It's a kind of electron pressure happening, and there's a clearly, if the ischemia is prolonging, a clearly long depletion of nucleotides because of the lack of the oxygen. ATP is breaking down, and that causes, if now reoxidation starts, that causes an acute overflow because the electron donors are immediately released, and something is happening in addition. There's a blockade of this wheel because of high proton motive force. That is, if I may say so, just a delay of the ATP synthase to have enough ADP. So ADP is breaking down, and therefore the wheel cannot circle. It's like you block the wheel and have, in addition, a huge amount of water which is coming to the wheel, and that causes an overflow in the wrong direction. It's like you have the mill and the wheel is stopped and you have a lot of water. So that is happening in the first second of reperfusion, and this is something which causes obviously complex 1 injury, that is known. So, and the most fragile location, which is also discovered in the last 2 years, I guess, is a small molecule sitting at the docking side of NADH. That is called FMN. That's actually a vitamin, phosphorylated vitamin B2, which is here placed as a prosthetic group. That's the first site of the electron donors, and that is fragile, and it can be released. If the electron pressure is too high in both directions, that can be released, and that causes probably oxidative stress. And this is something which injures complex 1. And so I think the message of these few slides is mitochondrial FMN release is probably very characteristic in many tissues for very initial reperfusion injury. And the story is that this is not a new message that has been discovered more than 30 years ago, also in hearts. So FMN loss is something which happens in the first seconds. And if we speak about machines which should prevent injury, then we speak about 3 different methods to avoid this complex 1 injury. One would be to avoid accumulated succinate, which is difficult because it happens always in ischemia if there's lack of oxygen, or to prevent mitochondrial loss. They're very effective mitochondrial scavengers, and I think, of course, that is feasible, but the scavenger has to go in the mitochondria at the right time, or we work on decrease rapid metabolization of succinates, something which that we avoid the rapid metabolization of succinate. And that is something very interesting because probably we can achieve that by just changing, for example, the temperature. And that's what we did. So we did an experimental setup and perfusing rodent livers, ischemic rodent livers with a perfusate at different temperature, at 37 degrees, at 10 degrees. And we we triggered the FMN release by giving succinate in the perfusate and increasing the succinate levels in the mitochondria. And you see what happens if we do that at 37 degrees. You can measure the FMN release by fluorescence in the perfusate. You see that we can trigger the release of FMN from complex 1 by giving succinate externally. And like visualization of the electron traffic jam, if I may say so. And if you do the same under hypothermic conditions, you see there's a huge difference in the release of FMN despite triggering the perfusate with succinate. And that was surprising, because there's obviously a difference in the temperature, in the conditions by complex 1 injury. And the message is there's less FMN or complex 1 injury in the cold. And was something new. We discovered that recently, and the reason behind that could be related to 4 things. Either we have a delayed AD transition, that the complex 1 is transferred from the actually dormating phase to an activated phase. That transition could be delayed, or there's a decreased proton motive force, or there's less reactivity of FMNH2, or there's actually inhibited RET. This is unclear, but the interesting thing is hibernators are also working like that. So they can decrease actually their proton motive force and therefore avoid reperfusion injury. So that may be a very ancestral mechanism to be protective, and therefore we think that one checkpoint for targeting machine perfusion is in fact mitochondria. And if we do that under cold conditions, that's actually something which is protective. So it may be smart to do before we introduce oxygen to ischemic tissue at normal temperature. It may be smart to do it a short period of time under cold conditions and afterwards reperfuse with normothermic blood. But this is only one side, because the next and maybe more important part is that we can measure, that we can quantify the injury of complex 1, because FMN is a natural fluorescent molecule. So in fact, what we are doing right now in Zurich is we perfuse with different machines, either normothermic or hypothermic, and we measure before we implant livers or also kidneys. We measure the release of FMN in the perfusate. And we decide, based on the measurement, we decide if we use the organs. So if it is major injury, like at a certain threshold, we discard the organ, and if it is less, we use the organ. I'll show you that in two practical examples. So let's say 2 DCD offers, 51 years and a relatively small functional warm ischemia time, a little bit fat. We put these organs on the Hope machine, hypodermic oxygenated perfusion, and the next is 54 years, also a functional warm ischemia time below 30 minutes, no fat. And the hepatectomy in this liver was delayed by a simultaneous lung retrieval. It happens sometimes, so hepatectomy was longer than 60 minutes, which is, of course, a concern. But you see here the livers, 2 livers, and macroscopically, I cannot decide which liver I would use. So I calculate the UK score, the risk score, which is the same. And so we don't know which liver would we use. And therefore we go for this measurement. And you see here the measurement. So in the first liver, with a very short hepatectomy time, the FMN level was very small, very calm. It was nothing happening, actually, like DBD, like control. And the other liver completely different, a steep increase of FMN in the perfusate. We checked that also by measuring NADH in the perfusate, and now we were interested, is that really something real? So we isolated mitochondria from both livers, from samples, and we found an 18-fold difference in mitochondrial FMN between those livers. And we were interested in addition on the proteins which are sitting around the FMN center. And this is actually something, there are 2 proteins in this N module, so-called N module, which capture the FMN, or where FMN is localized, and we can stain that. So we can stain that in tissues. And you see here, the one liver which we used had a nicely NDU-FS1 positive mitochondria. So basically, mitochondrial end module was intact, and the opposite in the liver, which we decided to discard. And we can also stain that by immunofluorescence. And you see here the staining of the implanted liver, which nicely show, like a control, by the way, which I can not show you that now for time reasons, but that is the normal picture of mitochondria in hepatocytes, and the opposite is happening in the liver, which we decided to discard, which look macroscopically the same. So this liver is completely black. It's like you switch off the light. So basically, the end module of complex 1 is severely injured in this liver. And of course, this data has to be validated. That's clear. And the pilot of a kind of preliminary validation is coming from UK. And see here, the proof of concept study suggested that FMN in the perfusate of kidneys and also of livers under normal thermic conditions, if you take the plasma, is also useful to decide if you use the organs or not. So I would summarize at the moment. I just can touch a little bit the perfusion principles because of time reasons. So at the moment, I think if we speak about oxidative stress, we have two options. Either we go for relatively small amount, short amount of perfusion and putting livers in hibernating conditions, or we go for very long perfusion, let's say 1 to 2 weeks, to have a fully functional graft. That's an opposite. So on the one side, we have a metabolic conditioning with ATP loading because complex 1 is not injured, and low succinate because it is metabolized. That will protect from ischemia reperfusion injury. And the other side is that we target regeneration, fat metabolization, maybe infections. So two different strategies. But the key of all the machine perfusion principles in the moment in the world is assessment of injury. That is something which is really a benefit of machine perfusion concepts. I will conclude. I think the metabolic status of the liver determines whether there's injury or not. The metabolic status of livers, which you cannot see, we need to measure that. We need to learn to measure that. And I think all oxygenated perfusion systems, also the hypothermic, they will trigger some injury during reperfusion, during reintroduction of oxygen, but of course at a smaller degree if you do that at hypothermic conditions. And the mitochondrial injury, which happens in the first seconds, that can be detected, and I think that's a major advantage to use it as tool for use or not during liver transplantation. Thank you very much.
Thank you very much, both speakers. This was very interesting. I think it was a good choice to have the two of you together in one webinar. So maybe we allow you first to comment on each other presentation before we go into the other questions. I'm interested in what you think of each other.
Oh, Martin, it's a long-standing issue, I think, and it's a major challenge ischemia reperfusion injury. And I think we are lucky to study a rather simple organ like the kidney, which has a clear clinical readout with DGF, which is much more complex in the liver. And I think one of the lessons I learned going back, but what we know now, going back to the literature, is that in the animal models, it's not clear whether we are studying ischemia reperfusion or ischemia reperfusion injury, because all if you look into the animal studies, they are very limited to a very short period following reperfusion, whereas what we've seen from our kidneys is that for at least half an hour, because our measurement window stopped, there is persistent metabolic collapse. And so there are so many challenges, I think, for us, and it would be very interesting to compare what we are seeing for the kidney to translate that to the liver.
Yeah. Thank you. So Philip, do you want to comment on that?
Yeah, I think it's very interesting what Jan said, because I think there's also a good connection of the two talks, because he said also, if I understood that correctly, that the metabolic situation is a problem. So you said also ATP depletion is something which you see much more than oxidative stress. And that may be a result of what we are seeing of mitochondrial injury. That for me, we speak always about mitochondrial function. So if there's no good function, we see, of course, a breakdown, and that's the disaster for livers. If there's no ATP delivery, there's no biflow, and then we have PNF, and that I think that could be the common line between the two talks. Yeah, whether it is in fact something which we cannot capture or which does not exist, that's very difficult to say, because, as I said, it's very difficult to look inside mitochondria, because they have very effective antioxidative systems. But what we can measure is, of course, damp signaling. So we measured a lot of damp signaling, which is downstream, of course, downstream. And I think that is something which we see, at least in the first minutes. We see a lot of damp signaling, and I guess that start in mitochondria. At least there are some interesting, actually from Holland, from Netherlands, interesting data about damp signaling, and in the first seconds, I remember nice papers there. What do you say about that?
Well, Philip, I think it's very interesting, but the problem is that we also see damps from living donors.
Yeah.
And
yeah,
and it's gradual. So they're all sustained damage.
Yeah.
And to make life more complex, we also noticed that there is a different impact of ischemia reperfusion injury, or DGF, on the type of donor. So it's DCD donors are relatively protected. So it's not only the injury, but also the ability to bounce back with resilience. So I think that we have to look into the whole picture, and I think one of the challenges is what's causing the problem, but that's providing us with such a short window of seconds, and whether it's oxidative stress, something else. But so far, all clinical trials with antioxidants failed in humans.
True, completely true. Yeah, it's a disaster. We cannot by adding something, we have no success.
Yeah,
that's true. Yeah, that's true. Yeah.
So I think our challenges are beyond the first seconds.
Yeah, true.
And
exactly.
how to cope with.
Exactly. Yeah. So there's a nice common line between the talks, you see.
Yeah, very good to hear.
Yeah, a huge amount of confounders. That's our problem in transplant business.
So there are a few questions coming in, and there's, by the way, a prize for the person that asks the most questions. So I can read out the first question. It's from Henri Leuvening. Question to Jan: is a redox exhaustion in other large animals such as pigs or dogs also the driving force?
Well, that's an excellent question. This is purely hypothetical. It's simply based on building the metabolome of DGF. And I think one of the challenges is that with DGF, we have a clear readout of ischemia reperfusion injury. And if you look into the models, we have never driven the models to an actual, let's say, DGF type of injury. So in between PNF and simple ischemia reperfusion. So I think that there are major challenges for us to go into more fundamental research and to start understanding what's actually happening. The more so since we don't see it in the small animals, and And cell culture is a particular challenge because cells in culture just switch off their mitochondria and rely on the glycolysis. So we can't study them.
Thank you.
Thank you. So the next question is both for you, Jan, and Philip. It's is there evidence that the pre-existing antioxidative capacity is important to protect against ischemia reperfusion injury, and are old donors more at risk? Would you like to start, Philip, maybe?
Actually, I wouldn't say that. I wouldn't say that, but of course I have no data about that. I just can say I don't see personally more oxidative stress in older donors when we, for example, measure surrogate markers. We don't see a correlation of the age alone. We see a strong correlation with warm ischemia, also with cold ischemia plus warm ischemia. We see also that the recipient condition matters a lot. So also the amount of vasopressors, for example, but we don't see something which is strictly related to age.
So could you say that it is in marginal donors also that you see it, or?
Of course.
In marginal donors.
Yeah, but you know, our problem is, as Jan said, what is a marginal donor? So who define what is marginal? Is that 65, 70, 90? Is that 30% macrosteatosis, 30% total? Is that functional warm ischemia of 30, 40, 20 minutes? Nobody knows that. Nobody defined that. I would say everyone knows it and everyone defines it differently. So that's our problem. The marginality is not defined, and therefore we have always different data. We cannot compare that. Yeah, so therefore.
Thank you. Would you like to comment on the question, Jan? Do you want me to repeat it, or you?
It's well, for me it's not such a complex question, because we don't see any oxidative stress. Also, if you look into the allantoin data, which the first sample was taken 30 seconds after reperfusion, there isn't any allantoin formation. So this tells me that there isn't any superoxide is escaping from the cells. So this challenges the whole aspect of how important antioxidants are. What is important, I think, is the resilience aspect with all the donors. A very remarkable finding we had was that the incidence of DGF is tripled in DCD donors, but we also found in the registry data that DGF in DCD donors doesn't impact graft survival. And when we looked into the molecular pathways, we found that in DCD donors, the resilience pathways are already activated at the moment of transplantation. So I think this also brings in a very interesting aspect, particularly with more marginal donors. How well are those organs able to cope with a period of energy depletion and stress, and how well are they able to recover?
Okay, thank you. There's another question coming in, and that question is actually to Philip. So the liver that was, you showed 2 livers as an example. So the liver that was not accepted based on the FMN levels. Do you think it will recover after 1 or 2 weeks of NMP?
Yeah. Who knows? So may happen. That actually may happen. We did that. You probably know it, that we perfused livers which were initially rejected for 7 days, and we had some recovery in some livers. And but the point is, we at the moment we never implanted these kind of livers up to now. We wait for that. So I think nobody knows what will happen if we perfuse at normothermic conditions livers for 1 or 2 weeks and implant then. This data is at the moment not existing, and I think it can come, but I don't know. I don't know. The answer is I don't know, unfortunately.
Thank you.
So Philip, can I just ask you, when you perfuse over so long time, is there something, do you think that it would be good to add, you know, like hormones or anything else during that time that would improve the graft?
Yeah, it's a lot. It's a lot which you have to add, because liver likes to be likes nutritions and a lot of things, but we are still looking for the best cocktail. I have to say. It's nothing specific. So from the hormones, we just, of course, we balance the glucose and the insulin there, but nothing special. Yeah. So I have at the moment, no secret drug which we can add and everything is fine. I have no.
Yeah. So some groups has looked at cytokine perfusion, you know, cytokine filtration during machine perfusion.
Yeah, of course.
Do you think that's an option?
We tried that.
Yeah.
But, so usually I have to say, if there's this burst of injury which we see actually in if we put livers on the normothermic device, so we see in the first hours suffering, clear suffering of grafts, like we see in vivo, like in the transplant business. So we see that, and we try to filter the cytokines, but that ended always in exchange of filters, because they are blocked by thrombocytes and coagulation factors, and so it's not very successful. The systems which are at the moment available may change in the future. Yeah.
Thank you.
I think
we have
a question. Sandra, do you?
Yeah, we have another question, and that is from Maria Kaiser, and it's to Jan. And the question is: are there evidence that different cell types in kidney have different thresholds of injury or different capacity of recovery, and how can that drive interventions?
That's a complex question. No simple answer. There definitely there appears to be a relationship with the energy requirements of cells and the susceptibility to ischemia perfusion injury. I mean, in the kidney, it's the distal tubuli which have the highest energy requirements and also are the most sensitive. And in the kidney, in the liver, the bile ducts have an extreme metabolic demand. This may also explain why they are the most vulnerable cell types. Could we target them specifically? Well, one interesting thing we found in our studies was that the kidney actually prefers middle chain fatty acids as the preferred metabolite, and that's also observed in pig studies. So as clinicians, we think simple in saline and glucose, and maybe we have to come up with a much better cocktail. So for the heart, the long chain fatty acids, which is preferred substrate for the heart. For the kidney, it's medium chain. And I think for the bile ducts, it would be very interesting to see whether they have a specific preference for metabolite.
Okay, thank you very much. I'm afraid time for questions is up. Sorry for the people that posted questions that we cannot answer, but maybe we can answer them another time. So there's a few more comments to make. So this webinar was recorded, and if you wish to look back, you can find it at the TLGA website. It's on screen now. Also, if you have any suggestions how these kind of webinars can be improved, you can add your comments in the chat box, which is also on the screen at the bottom. And then we would like to end with a very short poll, which will appear in the screen in a few seconds. And if you can please fill in this poll, it will be on the screen for half a minute about. Gives us a little bit of a break. Okay, so that's the outcome of the poll. Thanks very much for filling in. So then we're at the end of this webinar. So I would like to thank both the speakers very much, and also Sandra very much for co-chairing this with me. And then I wish everybody online a very nice evening, and thanks for tuning in.
Thanks to all.
Next time.
Bye-bye.
Thank you very much.
Bye-bye.
Bye.
Bye-bye.